Yeah, there are some when we are doing the research, we are not 100% sure that we will get the result. Because if we will know that we will get the 100%, we are not doing the research. Research itself saying that research means
Mm.
that searching every time. If you're not, although if you're performing the experiments, if you're not getting the result, then you have to research. Means that again you have to search
I
whatever
see.
the pros and cons and all this thing.
Mm.
So in my career, in my experiments of a biohydrogen, sometimes what happened that we get a low yield of hydrogen.
Mm.
Low yield of hydrogen means that whatever the theoretical value we are expecting to produce, so we are not getting that much of this biohydrogen from there. So it's a very challenging. Sometimes what happened that when we are
Mm.
scaling up, so whatever the experimental conditions we are providing to the anaerobic reactor, we are giving
Okay.
them a control pH, we are get control RPM and all this thing, nutrient balance in a small scale.
Okay.
When we are transferring this small scale reactor to a large scale,
Mm.
sometimes their productivity is decreased.
I
So
see.
these type of challenges we are facing, so
Mm.
we have to work on that. And second thing is that mythnogens. What happened that when we are using a hydrogen producer, it's
Mm.
not sure that only a pure type of bacteria is present. Their
I
type of
see.
some type of bacteria is also present, which
So,
is
so
known
you,
as yeah.
yeah, sorry, Dr. Singh.
Yeah,
So is
yeah.
there some kind of, are you saying that sometimes there could be bacterial contamination that if
Yes,
is
yes,
not,
yes. You are 100% true. Whatever, 100% true.
mm.
That there are some type of contaminants. We can't say that it's a contamination, but it's a
Oh,
competitor of a hydrogen production
I see.
producers. So
Mm.
what happened that there are some type of bacteria which is known as mythnogens.
Okay.
So they are a competitor of hydrogen. So what happened that all the organic matter which we are giving to
Mm.
the anaerobic reactor,
Mm.
whatever the hydrogen is producing by the hydrogen producers,
Mm.
so that hydrogen is utilized by the methane
I
producer.
see. In this episode of the Solomon Atah podcast, Dr. Neeraj Kumar Singh explores the science and scalability of biohydrogen production through microbial electrolysis systems. The conversation unpacks how wastewater, lignocellulosic biomass, and bioelectrochemical processes can be engineered into viable renewable energy solutions. Moving beyond laboratory success, the discussion focuses on industrial scale up, interdisciplinary collaboration, and the future of sustainable bioenergy in emerging economies. My guest is Dr. Neeraj Kumar Singh. He is an environmental scientist and researcher specializing in bioelectrochemical systems, biohydrogen production, and sustainable energy innovation. His work bridges environmental engineering, biotechnology, and renewable energy systems, with projects supported through national and BRICS research collaborations. He focuses on advancing scalable solutions that transform waste streams into clean energy resources. The Solomon Atta podcast starts now with Dr. Neeraj Kumar Singh. Welcome, Dr. Singh, and thank you for being on the podcast.
Thank you, Eta, for your invitation. It is my honor to be in your podcast.
Thank you. It looks like you are in a lab. What is fermenting right now as a biotechnologist? What are you looking
Yeah,
at?
yeah, yeah. I know it's a holiday, but I'm monitoring my reactors, which are producing the hydrogen.
Okay. What what
Yeah,
are what, what are reactors? Just explain that to me a little bit.
a reactor is a type of small vessels
Yeah.
where the reaction is taking place. So
Mm.
we give a optimized condition to the reactors, and
Yeah.
after that, in that optimized condition, or bacteria or any organism is producing our products which
Mm.
we are desired.
Okay. And what has been produced from that reactor before?
Currently, we are producing hydrogen from there and some of
Yeah.
the volatile fatty acid from it.
Oh, I see. That must be interesting, eh? I'm thinking
Yeah,
people that I mean, if we can hear it as ordinary people, but for you, when you are seeing all that reaction, surely it is making you feel good.
Yeah, of course. That's why I'm here. I'm here in this my lab just now
Mm.
to look at all this thing.
Okay. So what was the first thing that made you to choose biotechnology and biohydrogen systems, electrochemical processes? What was the first thing that make you like it?
Yeah, frankly speaking, when I was completing my master's, I was
Mm.
not having any idea of the research.
Okay.
But my mentor, Dr. Venkatesh Kumar R, he was a first one who was pioneered me, my interest to work in the biofuel production.
Okay.
So, but I was not knowing the area, which area I should work on that.
Mm.
So after that, when I joined to my PhD in
Mm.
Central University of Gujarat,
Yeah.
my supervisor, Dr. Rajesh Kumar Singh,
Mm.
he has motivated me to work on a biohydrogen production,
Okay.
because at that time he was thinking that if I will work on a biohydrogen, I will having a very good my career in the future. I will get a good chance to do as to become a scientist and to
Mm.
become a postdoctoral fellow. Easy for it will be easy for me to
Mm.
get the postdoctoral fellow. So
Mm.
he has boosted me that confidence
Mm.
to work in a biohydrogen production. And when I was reading the paper, due to his motivation and all this thing,
Mm.
so when I was reading the paper, so I came across that when I will work on a biohydrogen production, it is a demand of an hour
Okay.
of research, because I have started my career in 2014
Mm.
as a this as joined as a post this PhD scholar at that time.
Mm.
So at that time, this hydrogen was in a booming stage.
I see.
So I think there is a less amount of work has been done.
Mm.
And when you're talking about a bioelectrochemical system,
Mm.
it was also in a booming stage
I
at
see.
that time. So I thought that I should work because it is a demand. All the world is looking for a clean and sustainable energy,
Yes.
clean and sustainable energy. That's why I chooses this area.
Mm. Fantastic. So, Dr. Singh, your academic path spans environmental science, sustainable development, and bioelectrochemical research.
Yeah.
What first drew you to biohydrogen and the microbial electrolysis system?
Yes. That's a very good question, and it's very interesting to you to tell you that why I've chosen this bioelectrochemical integrated this bioelectrochemical system with a hydrogen.
Mm.
As I've told you in during my introductory statement that
Mm.
this when whole world was due to the increase in the population and all population and energy
Mm.
demand this fossil fuel on which the and all the energy is depend. So
Mm.
these fossil fuels are degrading. So degrading, when they are degrading they for the energy and all this thing. So
Yeah.
what happened that when they are used for the this energy,
Yeah.
so they are also deteriorating the environment,
Mm.
because they are releasing a toxic gas into the environment, just like a
Yeah.
SOx, NOx, and particulate matter into the environment.
Mm.
So what happened that when these toxic compounds are released into the environment, so they are very causing a very lethal effect on the human body.
I see.
So so there is a good chance how we can minimize the uses of the fossil fuel.
Mm.
So we can use as a this renewable resource for production of a
Mm.
hydrogen.
Mm.
So I use this renewable resources just like agriculture biomass and
Yeah.
this, and by integrating it with a bioelectrochemical system,
Mm.
because the bioelectrochemical system means that integration of microorganism, which is a bio, and after with the electrochemical system means that with the electrochemistry we can
Mm.
integrate it, and we
Mm.
can degrade this organic matter. We can produce energy, and we can produce this we can produce energy in form of electricity, or we
Mm.
can produce into the form of a clean hydrogen energy.
I
So
see.
that's why I've integrated this microorganism with a bioelectrochemical system.
Mm. Mm. That must be very exciting. I'm just thinking about it now as an ordinary person, that just to be able to see how all of these things are coming together and you getting biohydrogen out of biomass, out of some kind of waste. That must be an incredible, exciting process.
Yeah, it was a very incredible, because when I started my career, I
Mm.
was also not knowing whether I will produce my the hydrogen or like,
Yes.
but I was very happy to see when I got the first week when I was
Oh,
doing the
nice.
analysis. Yeah, in it's a very good experience when you
Mm.
perform the experiment for a 24 hours or a 48 hours,
Yeah.
and when you go for the analysis with gas chromatography, when
Mm.
you will see the peak of a hydrogen, it is very interesting at that time. So
I can
whatever the
imagine.
effort, yeah, whatever the efforts you
Mm.
have given for a 48 hours and all this thing,
Mm.
so that will be fruitful for you, that will be taken.
Mm.
Yeah.
Okay, can you just, Dr. Singh, explain how microbial electrolysis systems work and why they matter for renewable energy and wastewater treatment?
Yes. It's also very interesting to tell
Mm.
you about it. Generally, this if we say that how this works, so
Mm.
generally this bioelectrochemical system, as I told you earlier,
Mm.
that to the viewers, that this generally use a bacteria, and after that they use to produce they used to produce this bacteria. When integrated with a bioelectrochemical system, they used to produce electricity or this hydrogen.
Mm.
So generally this bioelectrochemical system works in three steps. Do you
Okay.
know the first step is use of organic matter. Organic
Okay.
matters means, just like in a wastewater, when we are discharging this our wastewater into the environment, they
Yeah.
contain a food waste, they contain a organic waste. So
Yeah.
what happened that these organic waste when they are put into a reactor, which is an anaerobic chamber,
Yeah.
so these bacteria utilize that organic waste. So what happened that they use the organic waste, so these organic waste is utilized, and after that they are degraded, and they produce a hydrogen inside it.
I
So
see.
what happened that in the first steps they use this thing, they degrade this organic matter in an anaerobic condition. Anaerobic condition means that the condition where oxygen is not present.
I see.
So yeah, when this oxygen is not this degradation of organic
Mm.
matter is done by a bacteria in absence of hydrogen,
Mm.
they produce a hydrogen inside it. And interesting, I want to tell you that when it will occur in an aerobic condition, so what
Okay.
will happen that whatever the hydrogen is formed into a reactor, so that
Mm.
will convert it into a water. The
Okay.
hydrogen will not form. So that is why it's a very key factor of hydrogen production during a dark fermentation process, that it
Mm.
will occur in a anaerobic condition.
Mm.
So after that, once this food in a wastewater,
Mm.
this organic waste is degraded. The second step is that
Mm.
they, when it's degraded, it's releasing ions. Ions means that electrons can be in raises, and
Mm.
after that, these electrons are migrated from anode to a cathode through an external wire,
Mm.
and they deposit on the other electrode captured in the third step on the cathode, and where the hydrogen or electricity is is produced over there. So
Mm.
basically, if we say into a simple word, that how it applies in a wastewater, so that we put a bacteria
Mm.
inside, or we give a waste to a bacteria, and
Mm.
this bacteria will do its job.
I
So it
see.
will do a job, and after that it will produce a electricity, this
Mm.
energy or a hydrogen from there.
Mm.
So they are using as a carbon source for the energy, and
Yeah.
after that they are producing a energy
Hmm. That's a
from there.
very sophisticated chemical
Yeah,
process going. But I just want
yeah.
to understand a little bit about how the absence of oxygen happens. Can you just explain that, Dr. Singh? How do you take away oxygen from those to produce that aerobic environment? How do you make oxygen to
Yeah,
disappear?
yeah, you are asking us such a good question. Even the scientist cannot ask this type of question, although
No,
you
but
are not from that.
I'm listening to
Yeah,
you
yeah, yeah, yeah.
because you just says
Yeah,
the
that's a
oxygen is not there, but then how
yeah,
do you make the oxygen to disappear?
yeah.
Yeah. so that
So
it can
as I
then
tell
give you the
you,
product. Yeah.
yeah, yeah. As I told you earlier,
Mm.
that whatever the reaction we
Yes.
are performing in a reactor.
Okay.
Reactor means a place where the reaction is taking place.
Place.
So what happened that in a reactor we are having a special pores from where we put a nitrogen inside it,
Yeah.
the reactor in the sea level. So whatever the oxygen is there inside the reactor, that will convert into a N2, nitrogen oxide, oxide of nitrogen. So
I
whatever the
see.
oxygen is, that will be purged, so purged out, and after that we seal it,
I
and after that
see.
seal it, yeah,
Then
in a
then
serum
the
bottle
oxygen is
way,
absent now.
absent. And
I
now
see.
the this bacteria will perform its
Is
activity,
job.
and they will use, yeah, its job, and after that they will degrade this organic matter and produced. So interesting, I forgot to tell you one thing, that
Yeah.
if we are using this biochemical system, we are getting a many benefit. What?
Okay.
That we are cleaning the wastewater, because this bacteria is degrading the organic matter of the wastewater,
I
and
see.
after that they are reducing the population also, because see, when this bacteria we use the organic matter in the form of a COD, so they
Mm.
will reduce this organic matter from there, so ultimately
Mm.
they are
Mm.
reducing the population, and whatever the wastewater we are treating into the environment, so uh, that is effluent we are escaping into the environment, so
Mm.
that is in a reduced toxic form,
I
so it
see.
will not deteriorate, and after that these waste, which is a wastewater, we are
Mm.
utilizing it for producing energy. So it is a very interesting for this thing.
Yes. I'm just thinking that someone's going to watch this podcast and start wondering whether can they come to your lab and see how this is? Do high school students come there to see this? Because I think that, I mean, just imagine this process being seen by high school pupils, students that will see it for the first time. I'm sure this is going to really stimulate a lot of, do you
Yeah,
have a lot of interest? Do you have
yeah, yeah.
I know maybe the undergrads will, of course, be there, the honor student or master students, of course. But what about high school? Is there something like that that kids come to your lab
Yeah,
and
due, yeah, yeah, our university used to perform some type of activities, so
yes,
they call the students from the nearby area, and
yes,
they told tell us to them to visit our lab, and
yes,
there they see the lab, they ask some multiple questions from
yes,
us, and after that I clearly see from their faces that they are so
yes,
much interested,
yes.
and that's a good thing which South African universities are doing.
Mm,
They are promoting the sciences on the basic scale of a high school,
mm,
so and they are creating, and after that when they are getting the opportunity to get admission in a DOT, and they are doing a
yeah,
wonders.
absolutely.
It's a good thing, every other country should learn from the South Africa.
That's incredible. I really like the way you say it.
Yeah,
Um, Dr. Singh, in your research, what have you found to be the most significant barrier to scaling biohydrogen production from lab settings to industrial application?
yeah, there are some, when we are doing the research, we are not 100% sure that we will get the result, because if we will know that we will get the 100%, we are not doing the research itself saying that research means
Mm,
that searching every time. If you're not, although if you're performing the experiments, if you're not getting the result, then you have to research, means that again you have to search
I
whatever
see.
the pros and cons and all this thing.
Mm,
So in my career, in my experiments of a biohydrogen, sometimes what happens that we get a low yield of hydrogen.
mm,
Low yield of hydrogen means that whatever the theoretical value we are expecting to produce, so we are not getting that much of this biohydrogen from there. So it's a very challenging. Sometimes what happen that when we are scaling up, so whatever the experimental conditions we are providing to the anaerobic reactor, we are giving
Okay.
them a control pH, we are control RPM and all this thing, nutrient balance in a small scale.
Okay.
When we are transferring this small scale reactor to a large scale, sometimes their productivity is decreased.
I
So
see.
this type of challenges we are facing, so
Mm,
we have to work on that. And second thing is that methanogens. What happened that when we are using a hydrogen producer, it's
mm.
not sure that only a pure type of bacteria is present. Their
I
type of
see.
some type of bacteria is also present, which
So,
is
so
known
you,
as yeah,
yeah, sorry, Dr. Singh.
yeah,
So is
yeah.
there some kind of, are you saying that sometimes there could be bacterial contamination that if
Yes,
is
yes, yes.
not,
You are 100% true. Whatever, 100% true.
mm,
That there are some type of contaminants. We can't say that it's a contamination, but it's a
oh,
competitor of a hydrogen production
I see.
producers. So
Mm,
what happened that there are some type of bacteria which is known as methanogens.
okay.
So they are a competitor of hydrogen. So what happened that all the organic matter which we are giving to
Mm,
the anaerobic reactor,
mm,
whatever the hydrogen is producing by the hydrogen producers,
mm,
so that hydrogen is utilized by the methane
I
producer
see.
medusa. So they are
Mm,
not using to make a hydrogen, but they are converting this methanogen is converting methane into a methanogen. This methanogen is converting hydrogen into a methane,
I
and
see.
when we are having a requirement for producing a form of energy, this
Mm,
methane is not a clean form of energy,
I see.
because this is a greenhouse gas. So
I
what is
see.
happened that this causing a, this methane is also causing a greenhouse, although it's give a energy.
Mm.
Methane is a type of the gas which is giving the energy, but it's not the environmental friendly,
Is
because
that
it
not the
will
one that we
cause
use at home, Dr. Singh? Is that is
no
methane
no
not
no
the
no
one?
no
Okay,
no
this
no
is
no
a
no
different
let's
form of methane.
yeah yeah.
Okay,
We are using in that this biogas plants and all this thing we are
I
using
see.
that.
Okay,
So this is not a pure form of gas.
okay.
So these, yeah, although some of the people are doing work on a methane, but when we are thinking about the sustainable environment
Mm,
and eco-friendly environment, this
yeah,
methane is not
not the good
will
one.
play not a good win, because it will causes a greenhouse gas and climatic change and all this thing.
Mm,
So we need that gas. We need a hydrogen as a clean form of energy. So
mm,
we have to innovate. We have to take a special precaution for this methanogens to innovate in.
mm.
And third is viability of a wastewater.
Okay.
Means that what happened that when we are this wastewater is coming from the different industries, and they are
Mm,
going to a specific place for a wastewater treatment plant for
yeah.
the treatment.
Yes.
So generally what happened that the organic content in the wastewater is not same every time. Sometime it
Okay.
will be low and sometime
Sometimes
it
it's
will be
more.
high. Yeah. So at that time what will happen that the hydrogen productivity will be less at that time. And fourth challenges, third and one of the main challenges, if we
Mm,
are not using a wastewater, we are using a plant just like a
mm,
agriculture biomass
yeah.
for production of a hydrogen.
Mm,
Sometimes every season, suppose we are producing a hydrogen from the sugarcane bagasse, so sugar bagasse is not every time available, because it's in a specific season, and when we require this biomass in for production of a hydrogen, it will be not available. So
mm,
this is also a key challenges. And
mm,
after that, sometimes inhibitors are also formed, so
mm,
during the pretreatment. So
mm,
this inhibitor is causing a many problem to our hydrogen producer. So they are reducing the at this productivity of hydrogen. And after that, the last one is economic feasibility,
mm.
because for establishing when we are transferring from the lab to a land, our
Yes.
technology, whatever the technology we have developed in
Yeah.
our this lab. So what happened then when we are transferring our technology to a
Yeah.
land. So what happened that it requires a lot of financial out financial budgets and all this thing,
Mm,
because it's a not a cheap process. So some type of subsidies should be provided by the government so that we can get revenue and provide jobs to our this
mm,
local people on this thing.
mm.
These are the
Okay.
important challenges.
So, if I may ask, so when you are as a researcher in a lab, how much quantity, like the highest amount quantity of hydrogen, have you been able to produce? Can you say 10 liters? Can you say 100 liters? Can you say five
No, no.
liters? Like, is it just a research with a small amount? What kind of quantities are you working with at the level of the
Yeah,
lab?
actually, we are working on a 5 liters still, because if we want to go above 5 liters
Yeah,
of reactor, we have to go outside. We can't perform our experiment inside our lab.
I see.
Okay, or we can perform our experiment in 120 ml serum bottles. So when
Mm,
we are going to the lab, to this, so we are generally producing in, if we are taking a reactor of this 120 ml, we are producing around about 50 to 60 ml of hydrogen from there.
I
But
see.
we can't say that it's a ml, but we actually measure it into a moles of a glucose. Means that
Okay.
if we are, yeah, moles means that if we are having giving 1 mole of a glucose, so how much generally we produce a 4 mole of a hydrogen from here, we
Mm,
calculate it from there.
I
So
see.
for getting that idea
Mm,
is unit till that date, we, I think, if we see that data or all over the world,
mm.
our group is a maximum producer of hydrogen from
Oh, great.
yeah, we
Yeah.
have produced around about 3.74 moles of
Mm.
hydrogen from 1 mole of this glucose till
Yes. Oh,
that
that's
day.
great.
Yeah, that's a
And that's
great achievement from it, and
Yeah,
it
and
has
that's happening
that
in Durban University of Technology. Is that what you're saying, Dr. Singh?
yeah, we are also
Incredible.
producing. Yeah, our group is also working on algae. They are using algae as a biomass, and from there they are producing a hydrogen. And we
Incredible.
are good, yeah, we are getting it. And due to this, our university has also decided to open an institute of hydrogen energy they have proposed in 2026.
Mm.
If we will get, so that is providing a lot of fund to
Yeah.
our, and they are supporting the young researchers of the South Africa and postdoc trend.
Mm.
They will support for this thing. So
Incredible.
they're targeting, yeah, this thing.
Great, because I just wanted to get an idea about what kind of quantities we're working with, and you've provided that response. So, Professor Dr. Singh, how do pre-treatment strategies for lignocellulosic biomass influence hydrogen yield in bioelectrical, biochemical. Sorry, let me take that question over. How do pre-treatment strategies for lignocellulosic biomass influence hydrogen yield in bioelectrochemical systems?
Yes, Atah. I want to tell you this thing that lignocellulose, for the viewer,
Yeah,
first they
what
should,
is that, by the way? Because that's a
yeah,
very fancy term.
lignocellulosic biomass.
Yeah.
Actually, this lignocellulosic bio means that cellulose containing a lignin, means that a agriculture biomass or a plant biomass.
Okay.
They are called a lignocellulosic, means that they are containing a lignin. That
I
biomass is
see.
containing a lignin.
I see.
We can simply say this thing that if we take a gift and we wrap our gift,
Yeah.
so that wrapper is called lignin. We
I
can say
see.
that wrapper means that
Okay.
plant biomass, that lignin wrap or biomass,
I
means
see.
that protect or this biomass for
Yeah.
degradation, for
Mm.
insect activities from predator and all these things, they
Yeah.
protect that. So they protect from the bacteria for
Mm.
utilizing that biomass. So
Mm.
generally what happened that now that lignocellulosic biomass is clear. So this lignocellulosic biomass is just like a crop. They can be straws of
Mm.
plant, they can be wood, they can be, so this is wood, and this wood is a complex structure of hemicellulose, cellulose and lignin.
I
As I
see.
told you that this lignin protects the outermost cover of a cell wall.
Mm.
So this lignin, so what happened that when we
Mm.
pre-treat it means that for giving bacteria this lignocellulosic biomass, this lignin has to be removed, so
Okay.
that can be eaten. by a bacteria to produce a hydrogen. So for that lignin removal, we use the pre-treatment. So what is that?
I
We
see.
pre-treat that lignin with this acid, means that we put our biomass inside the acid. So what happened, that that lignin layer will be dissolved.
Yes.
Okay, and
Removed
after
or
that
something. Yeah.
removed, yeah, removed.
Mm-mm.
So what happened, that this lignocellulosic, this lignin will remove, so it will liberate its hemicellulose and cellulose from there. So
I
what
see.
happened, that this pre-treatment, and after that when this is removed, this lignin, so
Mm.
now the bacteria will go and attack on this thing,
Yeah.
hemicellulose biomass, yeah. So it will be easy from them, so they will go and attack on this hemicellulose and cellulose,
Mm.
and so they will oxidize it, this organic matter,
Mm.
and now
Mm.
they will produce the hydrogen,
Mm.
hydrogen from air, or energy from air. So if we want to ask me a funny statement from me, that what is this lignocellulosic
Mm.
biomass, we can see that it's a tightened
Okay.
gift.
It's
So
something that you tie the
yeah,
gift in, and the
yeah,
gift is
yeah,
is the
yeah,
biomass
yeah,
material that is inside.
but
But you
yeah,
have to pre-treat it by removing or by
yeah,
dissolving the ligno in order for
yeah,
you to liberate the
yeah,
material that you need
yeah.
for your work.
Yeah, but point
Okay.
to be noted in this thing, that
Mm.
when we are pre-treating this lignocellulosic,
Yes,
sometimes what happens, sometimes a toxic chemicals is also related, which is
I
called
see.
the inhibitor.
Okay.
So we have to liberate this lignocellulosic biomass in an optimized condition.
Okay.
Means that, yeah, optimized, so that these inhibitors are not liberated from there.
Mm.
Okay, means that if you're opening a gift, that bacteria should not
Hurt
open
you or
that
something.
gift
Yeah,
too hard, so that it can harm you. So it need to be a precautionally liberated,
I
so that a
see.
minimum of inhibitor is formed inside it, so
Mm.
that it can be utilized it for the hydrogen production without inhibiting the growth of the microbes inside the
I see.
reactors. Yeah.
Interesting. Dr. Singh, you have worked on projects funded through national science agencies and BRICS initiatives.
Yeah.
How does interdisciplinary and international collaboration advance this field?
Yeah, true. Yeah, yes, yes, it was very honor for me to work in a different funding agencies.
Mm.
Uh, Probably before coming to the South Africa, I was working in a major government organization funded
Mm.
by government of India.
Mm.
So which is DST, Department of Science and Technology,
Mm.
and DRDO, Research Development Organization, and BRICS countries,
Mm.
and all this, and Department of Biotechnology. They have funded me a lot. They
Mm.
have funded me a lot in this field to work
Mm.
on a biohydrogen,
Mm.
on a bioethanol, and this and development of develop wastewater treatment, and development of an anaerobic, and it has helped me a lot. It has motivated me to collaborate with different countries,
Yeah.
different different countries all over the world, just like Brazil, Russia, India, China,
Mm.
and South Africa. So
Mm.
they have, I went there, and after that I have learned the latest technologies and
Yeah.
methodologies which they are, and they have also learned from me some of that
Mm.
during the exchange program and all
Yeah.
this thing. They have also learned something from it, and we are totally collaborating. If
Mm.
some problem we are facing in our research, they are helping us, and if they are facing some problem in their research, they are also helping our, and they have also supported me with
Mm.
some of that international conferences and all this thing, so
Yeah.
that I can easily go any part
And
of the world to
yes.
present my research, and after that I can collaborate with the different scientists of the world.
Yeah.
And after that, this South African government is also financially supporting us, our team here, Prof.
Mm.
Santosh Pillai, and our research group in
Mm.
establishment of this lab,
Mm.
buying this advanced instrumentation, just
Yeah.
instrument just like a gas chromatography, which is
Mm.
analyzing the hydrogen,
Mm.
analyzing the hydrogen, HPLC, and all this thing. They
Mm.
are supporting me as a lot. to carry out research on the biohydrogen production.
Mm.
And as I told you earlier, that they are also supporting us to build a new institute of a hydrogen energy and duty.
I
So they
see.
are supporting us a lot, so that we can give them such a technology
Mm.
that can empower the people of South Africa.
Fantastic. And the global south as a whole.
Yeah,
So
yeah,
that is wonderful. Dr.
that's
Singh,
true.
as we start to wrap up, I still got a couple of questions for you. What intersections do you see between environmental engineering, sustainable energy, and biotechnology in Africa research landscape?
Yeah, yeah, it's a yeah. See, what is there is that in Africa, whatever the huge amount of agricultural biomass is there, huge
Mm.
amount of agricultural biomass. If we take the example of the world around, nearly when we see the latest, I think some of the latest data is still unpublished over here.
Mm.
So when I was reading the paper, I found that in all over the world recently, around 5 to 7 billion metric ton of biomass has been produced. So which
Okay.
is unutilized.
I
After
see.
harvesting the field, it's unutilized, and so they are generally left in the field. So left in the field all over the world. So
Mm.
they are simply left, and after that they decomposed, and after that this is converted into a organic matter, and after that they fertile the land, but
Mm.
it's unutilized.
I see.
So what happened that if we take the all the African continent, so around 700 to 900 million tons of biomass is there. And if we take about the Africa, around 40 to 50 million tons of biomass is left, which is mainly a
Mm.
sugarcane. So it's a vast amount of lignocellulosic biomass. So we are working, we can use it for the production of hydrogen if by using this organic matter,
Mm.
this agriculture biomass.
Mm.
So and another thing is that in South Africa we can produce, and after that this the world is in a age of that they have a target
Mm.
of net zero carbon emission
Yeah.
of 2005. They don't want to emit any carbon into the environment.
Mm.
How much it's produced, they don't want to. So the government of South Africa is doing wonders in the field of South Africa, because they know the importance of green hydrogen production.
Yeah.
Means that green thing, because after 10 to 15 years it will be the era of, it's the era of hydrogen
Mm.
energy. But and after 10 to 15 years, all the vehicles and trains, aeroplanes, trains and all this thing will
Mm.
be fully operated on
On
this
hydrogen
hydrogen
biofuel.
energy.
Mm.
So the government of India, the government of South Africa is knowing this importance of this
Mm.
hydrogen. So they are launching a various programs, and
Yeah.
they are investing a huge amount of money. So they have launches a various program, just like a national green hydrogen program.
Mm.
So in this program, what is happening that they have invested around 800 billions rupees rands
Mm.
to carry out the research. So they are targeting about 13 million tons of this hydrogen in
Mm.
2050. They
Okay.
are targeting to produce this much quantity of hydrogen. And other
By
is they
2050.
are,
Yeah.
yeah,
Mm.
they are also integrating some of the project, fast track project, and project with the industries.
Yeah.
So they are also investing a lot, so that all that industries are operated on the hydrogen, so that they do not utilize the fossil fuel, and which is
Mm.
causing the environment pollution. They are also working on this field. This is also a very wonderful project. I think
Yeah.
they are learning from China, they are learning from the US and all this thing.
Mm.
This government is learning, and after that they are also creating a large ammonia production clusters in the Eastern Cape. They are making.
Mm.
So this is also a very good program, and after that they are also making a green hydrogen plants on Western Capes. They are producing,
Mm.
and they are also creating a hydrogen valley. Hydrogen valley means that they are creating a town or
Yeah.
any organization on which, where the hydrogen will produce, there will be utilized, and after that they
mm,
will perform all the energy will be given by the hydrogen. They are
mm.
creating it. So in 2000 they are creating a hydrogen valley in Johannesburg. They are creating a Richards Lake, and after that they are creating in Limpopo.
I
So
see.
they are creating, yeah, they are creating a lot. So
Mm,
their plan is that they want to sell this hydrogen in 2030 at dollar $4 and all this per kg.
mm.
So there
So
is a
and
lot of
oh,
investment.
yeah, all of these systems, are they linked? Are they're talking to each other? All of these places you've pointed, are they linked? Are they being coordinated from, or are they talking
Yeah,
to each other?
yeah, they are talking to each other.
Mm,
So because they are only funded by the government, they are
yeah.
funded, and they are supporting a lot from this.
Mm,
And another thing is that one of the plant, green
yeah.
hydrogen pilot plant, is also launched in this Johannesburg.
Mm,
So it's also reducing they are also they will be operated, I think, soon. I don't know the exact
mm,
news about this thing, but
mm.
they are working on this thing. And after that, the BRICS countries programs is also these, all the BRICS countries,
Yeah.
Brazil, Russia, India, and South Africa.
Mm,
They are also sharing a common platform to work on a biohydrogen production. So they are
mm.
sharing to reduce the utilization. They are sharing, they are working on this platform that they have to reduce this fossil fuels.
I
They
see.
have to reduce the price, and they have to emit net zero carbon into the environment. And
Mm,
after that, they have to promote the industrialization by using this hydrogen. And
mm.
after that, they have to create
Yes.
jobs. They have to create a money and a wealth from there.
Mm.
So it's a very good programs which they have launched.
So, I have a few questions more, maybe one or two. Looking forward, Dr. Singh, what novel research directions or technologies excite you most in renewable bioenergy and microbial electrochemical systems?
Yeah, to be very honest, I want to tell you this thing that the this thing is one of the emerging trend, which is
Okay.
known as the integration of bioelectrochemical with biorefinery.
Okay.
Very less amount of work is done on this recovery of value-added product.
Mm,
Value-added means that
okay.
with the hydrogen, other products are also released into
Okay.
the reactors. So if we recover that reactor, that will also definitely cause reduce the profit and the cost of our this this cost of our operational cost of the our technology. So
Okay.
we have to work on this thing. And after that, the second thing is that we have to identify a microbes, genetically modified microbes, so which can selectively inhibit the methanogens inside the reactor, which is a very challenging. Or we can
Mm,
use some of the technique to inhibit the methanogen, is also a very innovative topic.
mm,
And after that, third thing is advancement of the electrode material. Means that the electrode which we are using in bioelectrochemical system,
mm,
that should be modified. Means that it should be doped with a heavy metal, that type of metal which increase our hydrogen production rate of this bioelectrochemical system.
mm,
That work is is very important. And
mm,
after that, scaling up technology,
mm.
because as I told you in the discussion, that scaling up is a very challenge.
Yes,
Because whatever
because of
the
the cost of,
yeah,
yeah.
cost and on all this thing, if the
Mm,
subsidy just like South African government is giving, that is given across the world,
mm,
so that will reduce the cost. And after that, that will be the real utilization of the real waste,
mm,
real utilization. And so this is the way, very interesting thing.
mm. But just let me just ask a follow-up question a little bit. When you say that when you are getting the hydrogen, there's some other byproducts that are also
Yeah,
useful. What kind of products are those?
they are fatty acid.
Okay.
Means that when the glucose is degraded, they are also producing some fatty acid, just like
Mm,
acetic acid, butyric acid, propanoic acid, and
mm,
all these things. Sometimes ethanol is also produced inside it. So
mm.
they are also having a very good economical value.
I see.
So when you are setting up your industry of a hydrogen, so
Mm,
these product can be also he also produce with that, because they are very costly into the market.
I see.
So if when someone is establishing the industry, so
Mm,
they can use it as a other
mm,
byproduct, so that they can boost the economy of this uh, deer industry.
mm.
So
I
this is
see.
also a very good option.
Means if you solve one problem to produce biohydrogen,
Yeah,
you can get a whole lot of industries
yeah.
and commercialize
Industries.
products from it, from
Yeah,
just that one main focus of biohydrogen. That's
yeah,
incredible.
yeah.
Dr. Singh,
That's it.
we're starting to wrap up. What areas of all your work excites you the most? I think I've asked this question, but I just want to hear maybe one or two things. And what areas have we not discussed that didn't come into our discussion?
Yeah, some of the challenges is not covered, whatever the challenges, and
Mm,
the thing is that scale up technology is not uh, well understand.
mm,
So that need to be discussed more. And after that, funding and all this thing,
mm,
because until and unless you will not get the funding from the government organization or any institute, you can't work it.
mm,
Because at the end of the day, people will see the product, but they don't see the effort, how much effort the scientists are putting
mm,
to produce that products. So that requires a government investment inside it.
mm,
So if we will get that investment, we can do a marvelous in this field.
mm. That's why I want scientists to come to the podcast because the public also wants to see and hear what the scientists are doing in their labs. So when you are on the podcast like this, you explain your research, you explain your work, the public can appreciate. Even
Yeah,
some policymakers, even some government people can see and say, oh, this is a kind, even I know government is aware, but I'm
yeah,
saying the public, especially the public, can start to appreciate the work that goes on in research labs, in research institute, and the kind of work that people like scientists like yourselves and researchers
yeah.
like yourself do. So, Dr. Singh, what did we leave out from our discussion? Is there
Uh,
something we didn't talk on?
uh, what? Yeah, we have discussed all thing area.
Mm.
We have
So
discussed
what
all
are you
thing
going to,
area.
after this podcast, what are you going to be doing? I know you were looking at the fermenting reactors when we were starting. After the
No, I have.
podcast, what are you going to be doing in your lab?
Yeah, I will. I have to submit one of the article
Okay.
inside that journal. We
Mm,
have completed that work and we have written all this thing.
mm,
And for submitting that written, I will go for. I have written all this thing and I have to submit this that into a journal,
mm.
so that it can be readable to all that to other scientists who are working in this field or research.
Okay. And then
So
after that,
they
what
want.
what next? What is your next frontier?
Uh, my next frontier is to build my own company in
Yeah.
this hydrogen production,
Mm.
and if for that I have to learn more technologies.
Okay.
So I am working in that, and if I will not, then I'm satisfied with the scientist. But I will take one chance
Absolutely.
to build my
Dr. Singh, give me
skill.
2 or 3 names of people that have inspired your work, your research.
Yeah. Uh, One of the most thing which has inspired is my supervisor, PhD supervisor,
Yeah.
who is Dr. Rajesh Singh from BBA University. He's a
Mm,
professor over there.
mm,
So he has motivated me a lot, because when I have started my this experiment,
mm.
he was only the person who has always motivated me too. Because see, when you are doing the experiment, you are not sure that you will get the result. But when
Yes.
I was not getting the result, I used to go to him and I saw proof it's not coming. So
Yeah.
he used to say that no issue. If it will not come, where it will go. So he was a total motivator. And after
Yeah.
that, here my host Prof. Santosh Pillai, he is also very motivated
Yes.
person, and he is very motivating in every aspects, and
Mm,
every aspects in the funding and all this thing,
mm.
whatever the requirement we are in, he is providing
I see.
with
Great.
writing the projects and all this thing.
Dr. Singh, thank you so much for being on the podcast.
Yeah, thank you, thank you, thank you.